Okay, good morning. We're going to go ahead and start now. Anybody have any questions on anything? Okay, so we're going to go continue in looking at our muscles. And the last thing we were talking about in here was this slide here showing us movement of the action potential across the cell membrane. You know, we talked about how we get an action potential. We talked about depolarization, talked about sodium rushing in at the motor end plate. You know, we have an action potential, a nerve impulse comes down a motor nerve, it goes down to the axon terminal, it opens up voltage gates for calcium, which allows a neurotransmitter to drift across the synaptic cleft and land on the motor end plate, opening up sodium gates. Those sodium gates allow sodium to come in and cause a localized depolarization event on the motor end plate. That's not, that's not the action potential because we have to hit threshold. But that's how we get an action potential from a motor nerve to the muscle cell. Now we have to get the action potential spread across the cell. We may not even have an action potential yet. We have to depolarize the motor end plate to hit threshold. Now, our cell membrane is got a charge on it. The cell membrane has a charge of the muscle cell has a charge of minus 90 millivolts. All muscle cells have a charge of around minus 90 millivolts. So to get the nerve impulse, the action potential to spread across the entire cell, three things have got to happen. We've got to change the charge on the motor end plate to hit threshold. Now, threshold for a muscle cell is minus 50 millivolts. So we got to go from minus 90 to minus 50. We get from minus 90 to minus 50 by letting sodium rush into the motor end plate area on the muscle cell. It will cause a localized depolarization. If we hit threshold, then we will turn on voltage gates that are on either side of the motor end plate. And our depolarization will spread. The action potential will spread all the way across the muscle cell and down into it, immediately followed by a repolarization event where we put the charge back where it belongs. So let's show you how this. The excitation phase is very important. We got to excite the motor end plate. We got to get the threshold. The threshold in muscle cells is minus 50. The membrane, the resting membrane potential on a muscle cell is minus 90. So we got to get from minus 90 to minus 50. We got to see a change in the charge on the membrane of 40 millivolts. We see what happens here. This is when we see the animation there, we see the gate is closed. The gate is closed at minus 90. It opens up, when we hit threshold, it opens up at minus 50. These voltage gates are on either side of the motor end plate. The motor end plate gates, the sodium gates at the motor end plate, are chemical gates. They open when acetylcholine lands on them. The gates on either side of the motor end plate are voltage gates for sodium. They only open when the charge gets to minus 50, and they'll stay open until the charge gets all the way up to plus 30. So a lot of sodium's coming in and changing the charge in the membrane. And we haven't even talked about the potassium gates yet. So we got to hit minus 50. We hit threshold at minus 50. The fast sodium gates open up. Now they'll stay open until we get to plus 30. At plus 30, they will close and the potassium gates open. Once we go from minus 50 up to plus 30, that's when the sodium gates get to work. But at plus 30, the sodium gates shut down. Again, they're a voltage gate. At plus 30, the potassium gates open, and they will stay open until we get all the way back down to minus 90. They're not going to be open at the same time. Okay, so while this is not an animation, it does show you everything that's taking place here. We have the resting potential of a motor end plate is normally minus 90 millivolts, but when acetylcholine drifts across the synaptic cleft and lands on the sodium gate, the chemical gates for sodium on the motor end plate and opens them, sodium will rush in and they will cause a localized depolarization, meaning the charge on the motor end plate will drop from minus 90 to minus 80 to minus 70 to minus 60. Oops, nothing happens at minus 60. Nothing happens. Nothing happens at minus 70. Nothing happens at minus 55 or even minus 51. But when we hit minus 50, then the sodium gates will open, the ones that are on either side of the motor end plate over here. This is a sodium gate here. I don't know if they're purple or not, but there's a sodium gate. It will open at minus 50. It will let sodium flood into the sarcolemma and across the sarcolemma into the sarcoplasm of the muscle cell. It will stay open until we get to plus 30. The potassium gate's closed. As soon as we get to plus 30, the sodium gate closes and the potassium gate opens and potassium leaves the cell. And we're repolarizing. We're putting the positive ions back in the outside and the negative ions on the inside. So the end plate potential is the change on the charge of the motor end plate. So, it's sort of repeating myself, but acetylcholine released from the axon terminal binds the receptor sites of the motor end plate, opens up the sodium gates, and we change the charge on the motor end plate. It's a localized depolarization. It doesn't mean we're going to get a muscle contraction. It doesn't even mean we're going to get an action potential. It means that we are depolarizing the motor end plate. If we're lucky enough to hit threshold, and we usually do, if there's enough acetylcholine, then we'll get an action potential. So we change the charge on the motor end plate. So we're changing the end plate potential. So the depolarization, when we hit threshold, we have now created an all or nothing event. Threshold, the voltage gated channels in the cell membrane are going to open. And they're not going to close until they get to plus 30. This phenomena of an action potential, a depolarization wave spreading across the muscle cell membrane, will not stop until it's done. It is unstoppable. And when you have an action potential spreading across a muscle cell, you will get a contraction of that cell. As the depolarization wave spreads across the muscle cell and causes more and more sodium gates to hit threshold, and it will depolarize that region and the region next to it and the region next to it and the region next to it, spreading out from both sides of the motor end plate. That's the depolarization event. Repolarization occurs as the action potential is spreading across the muscle cell. As we depolarize a region and get up to plus 30, the sodium gates shut. And then the potassium gates open. And sodium goes outside of the cell. And now we are reestablishing the charge on the membrane. We want to have positive charges on the outside and negative charges on the inside. The cell doesn't care that they're putting potassium out there to reestablish the positive charge. It'll fix that later with the sodium potassium pump. But for right now, it wants to repolarize itself with potassiums, with positive charges outside and negative charges inside. That's what we do. So depolarization depends on whether or not an action potential occurs. Depolarization of the muscle cell depends on how many channels were opened by acetylcholine. Did we have enough acetylcholine? If we had enough acetylcholine to open up the sodium channels on the motor end plate that we hit threshold, then we are going to run and run and run. We will run through the entire, that depolarization wave will run through the entire muscle cell until it's done. There's no such thing as a partial depolarization or a weak depolarization or a strong depolarization. They're all the same. The action potential that's formed is exactly the same every time. Once we hit threshold, we're going to move. We're going to get a contraction. Now, again, to review this, what is depolarization? This is a, this little summarize it for you. Here we see depolarization, sodium coming in, repolarization, potassium going out. It is all or none. It either does it when we hit threshold, or we have to hit threshold. If we hit threshold, we're going to have a depolarization event. We're going to have an action potential. If we don't hit threshold, then it doesn't matter. We didn't get an action potential. But once it starts, it will run until it's finished. It will not die out halfway through. It will run until it's done. So, and to see how quickly this is, how short a time frame this takes, or how quickly this occurs, take a look at this chart. We see that the sodium channels open somewhere. Here we are between 0 and 5 milliseconds. Let's say that's about 3 milliseconds here. Our sodium channels are going to open here thanks to acetylcholine. We depolarize the motor end plate, and we go from somewhere along in here. Well, actually, we depolarize the motor end plate. So this is probably minus 50 here. So we must be, we're depolarizing here. The acetylcholine is already acting on us here. So we go all the way up to plus 30. Sodium's rushing in. We're changing the charge on the membrane of the muscle cell on either side of the motor end plate. Once we get up to plus 30 though, the sodium gates shut. The sodium gates in the muscle cell membrane are designed to open at minus 50 and close at plus 30 millivolts. The potassium gates open at plus 30 and shut at minus 90. So this whole thing lasts about, the whole depolarization and repolarization event lasts for about 7 milliseconds, which is faster than we can comprehend. Because to us, a muscle contraction is instantaneous. We can't comprehend 7 milliseconds. That's 7 thousandths of a second. So the resting potential is minus 90. Threshold is minus 50. If we hit minus 50, we will go all the way up to plus 30, and then we will repolarize back down to we will repolarize all the way back down to minus 90. So we can do it all again. So, okay, so now we know what an action potential is. We know that an action potential is a nerve impulse, comes down a motor nerve, is transferred to a muscle cell. Sodium rushes into at the motor end plate, causing a localized depolarization, which if it hits threshold causes an overall depolarization wave that spreads across the muscle cell. We've got us an action potential. Now, how does that get us to a contraction? I said, I said it did, but how do we get to a contraction? Well, the next step is called excitation contraction coupling. Oh, I said contraction. Now, this excitation in the muscle cell is the action potential going across the cell membrane. We've got our action potential finally. We hit threshold. Now we've got an action potential. It's spreading across the cell membrane. It's going down the T tubules. It's contacting every sarcomere in every myofibril of the muscle cell. The action potential gets down into the triads, the T tubule and the terminal cisterns on either end of the sarcomeres. And that action potential causes those triads, the cisterns, to open up and release calcium into the sarcolemma, into the sarcoplasm. Remember, the sarcoplasmic reticulum is containing lots and lots of calcium. They're the sarcoplasmic reticulum are essentially in sealed tubes. Well, we open them up. The action potential allows us to open up the sarcoplasmic reticulum at the terminal cisterns and allow that calcium to flood across the muscle cell itself. It floods across the actins, it floods across the myosins. So we have lots of calcium now invading the muscle cell. This is the purpose of the action potential. The purpose of the action potential is to get down to the T tubule level and open up the terminal cisterns. This is the purpose. This is the purpose. Let me stop this and bring the camera over to, is it this camera on or this one? Must be this one. So here we have our little model again. And let's bring it over. Okay, here we go. This is the blue things are the T tubules. These are the myofibrils. You can see the sarcomeres are these areas here. There's a Z line and there's another Z line. So that's a sarcomere. And we see the T tubules along in here. And we see the sarcoplasmic reticulum in here. The action potential will come from the surface of the cell membrane up here. Here's where the motor end plate's located, right up here. The action potential will form at the motor end plate, adjacent to the motor end plate when we depolarize, and it will spread down through these T tubules and these T tubules until it comes in contact with every sarcomere in the muscle cell. And these terminal cisterns here and here will release their calcium, and it will flood across all of these, all of the myofibrils in that particular muscle cell. So, and the calcium is what's going to allow our contraction to take place. So let's get back to, it is the calcium that allows the contraction to occur. So everything we've done up to this point, the action potential coming down the motor nerve, the action potential causing in the motor nerve, causing the release of the neurotransmitter, acetylcholine drifting across the synaptic cleft, landing on the motor end plate, opening up sodium gates for a localized depolarization that has hit threshold, which opens up voltage gates on the cell membrane of the cell, which allows sodium to rush in and cause an ongoing depolarization wave that runs through the entire muscle cell, otherwise known as the action potential. The action potential comes down the T tubules and opens up the terminal cisterns and allows calcium to flood the cell. Everything we've done to this point is to get calcium to be released into the cytoplasm of the muscle cell and cover the actins and the myosins. So let's see if this works for us.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within the muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so we see that the whole point of everything to this point has been to get calcium to be released. The calcium levels, calcium is high inside the sarcoplasmic reticulum, and when we flood the sarcomeres with calcium, we can get a contraction. Now, how does this happen? Well, the action potential goes down to T-tubules. We open up the gates for calcium. Calcium gets released. The problem with our actins and myosins is that the actin, the thin filament, is also wrapped around by tropomyosin and troponin. The binding sites on the actin for myosin, where we form what we call a crossbridge, are blocked by tropomyosin. That means the myosin heads that are on the thick filament can't grab onto the thin filament, because contraction is all about the thick filament yanking the thin filament towards the middle of the sarcomere. So the muscle doesn't contract. The muscle doesn't contract at all. We've got to get those tropomyosins out of the way so the myosin heads can grab onto something. So, and we're going to, so the key here is calcium. Everything we've done to this point is to get the calcium released into the sarcoplasm to flood the cell. Because what calcium does is that calcium binds to troponin. The troponin is the other protein in the thin filament. You've got the actin and you've got the tropomyosin, which is sort of woven through the thin filament, covering the binding sites, and then you've got troponin. Troponin is attached to the tropomyosin, and when you flood the sarcomere with calcium, the calcium will bind to the troponin, and the troponin will move. It will roll over. As soon as it gets, it's like it gets heavy or something. But the physical junction of the calcium to the troponin, where they bind together, will cause the troponin to roll over, drag the tropomyosin with it, and expose the binding sites. And that allows the myosin to actually grab onto the actin and form what we call the crossbridge. The crossbridge is the connection of the actin and the myosin. Every actin molecule has a binding site for myosin, and the myosin heads all have a binding site for actin. And they come together. And when they come together, they will automatically, the myosin heads will automatically fire and pull the actin on both sides of the sarcomere towards the midline. And as long as there's calcium present, the myosin heads will keep grabbing onto the actin and giving it a yank and giving it a yank, as long as the calcium is present. Once we no longer have an action potential, once we no longer receive action potentials, we will start pumping the calcium back into the sarcoplasmic reticulum and the contraction stops. So we have 4 steps. First thing is we've already got calcium out there now. Calcium has flooded the sarcomere. It has bound itself to troponin, causing the troponin to roll over and drag the tropomyosin with it. The myosin heads, which are already energized by ATP, the myosin heads will reach up and grab onto the actin. And as soon as they do, they're going to pivot towards the midline of the sarcomere. So the myosin heads on each end of the thick filament are going to all pivot towards the middle of the sarcomere. And by doing so, they're going to drag the actin they've attached themselves to with it. They're going to drag the actins towards the midline. That's the power stroke. Now, if there's ATP present, the myosin heads will let go, drop down, and then they will recock into their energized state again, reach up and attach to the actin again and fire. So we have, as soon as we expose the binding sites, the myosin head, which has been energized with ATP, will reach up and grab onto the actin. Then it will pivot towards the midline, dragging the actin with it. Now the myosin doesn't move anywhere except its heads. But when it attaches to the actin and pivots towards the midline, it will drag the actins along. The actins will be pulled towards the middle of each sarcomere. The sarcomeres will start to shorten. If there's more ATP available, the ATP will attach itself to the myosin head. The myosin head will let go. It will drop down. It will recock itself so it can reach up and fire again and have another power stroke. The only way it can do that is if there's ATP present. So.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the Crossbridge Cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules and thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1, cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3, crossbridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the crossbridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Crossbridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Now remember though, that it is the muscle fiber, the muscle cell, not the whole muscle, because we don't know how many muscle cells are being stimulated with an action potential. Many times we will use a variety of cells in our muscle to get things done. We're not going to use all the muscle cells in one fascicle to do something. We'll spread our demand on the muscle cells across the entire muscle. It depends on what we're trying to do. Okay, so let's take a look at this. Contraction. We know that tropomyosin gets in the way. Tropomyosin, the blue band there is blocking all the binding sites for the myosin heads. The myosin head can't get there. Now the myosin head is pre-cocked, ready to go. It's got the high energy ATP the high energy ATP has already given its energy to it by hydrolyzing to ADP and inorganic phosphate. It's ready to fire. But it has, it's in its cocked position. But it can't do anything because the myosin heads are blocked from the actin. But calcium lands on the troponin. The troponin rolls over and drags the tropomyosin with it. The myosin head can reach up then and form a cross bridge and give its power stroke and yank the actin towards the midline of the sarcomere. The myosin is going to kick out the ADP and the inorganic phosphate. The myosin head has, once it's attached itself to the actin, it kicks out the ADP and inorganic phosphate so it can accept another ATP. The only way it will let go is if it accepts another ATP. As the myosin head has grabbed onto the actin, given it a yank, and now it needs another ATP to let go of it, drop down and recock itself. And so we've pulled the thin filament over top of the thick filament. It has slid across, is that right? Sliding filament theory, sliding filament model. It has slid across the top of the myosin. The sarcomere has shortened. Every sarcomere in that cell shortens at the same time, because all the action potentials in that cell the action potential in that cell is causing all the sarcoplasmic reticulums throughout every myofibril in that cell to open up and bind. The calcium will come out, bind to the troponin, exposing the binding sites. So every sarcomere is shrinking, shortening up. The muscle cell is contracting. Doesn't mean any other cells around it are contracting, but this particular cell is contracting. Now the myosin will stay attached to the actin until another ATP comes along. That will allow the ATP lands on the myosin. The ATP is hydrolyzed. That the energy allows the that allows the myosin head to let go and recock itself, and then reach up and grab onto the actin and fire. So there's our cross-bridge formation you see here in the animation. You can see the myosin heads are in purple, and the binding sites are those little yellow dots on the actin. And the myosin heads will reach up, give it a yank, drop back down. Takes ATP for it to let go and drop back down. Now, half the muscle, half of the myosin fibers are in the process of firing the power stroke. The other half are reloading. So at any given time, half in each thick filament, half the myosin heads are pivoting towards the midline. The other half are dropping down to reload and do it again. And as long as we have calcium to keep bound to the ATP, as long as we have calcium to bind to the troponin, and as long as we have ATP to bind to the myosin and reload its power stroke, then we're going to contract. As long as we have both, as long as the calcium is available, as long as the ATP is available. So, and here's a nice slow sequence to watch this. You can see the the myosin heads are attached. Here comes the ATP. It allows them to detach. And, when they detach themselves, then they automatically go into their cocked position. There they go. And that will cause them to reach up and bind to the actin and pivot towards the midline. These particular myosin heads are up here in this area here. So they're pulling towards the left. If we were looking at these myosin heads here, they'd be pulling towards the right because we want to pull the actins toward the midline. So the Z lines come closer together, the entire sarcomere shortens. But we got to have calcium. You can see where the calcium up here in the big green circles, that's the calcium, but it has bound to the troponin, causing it to roll over and drag the tropomyosin, those white bands, out of the way. The binding sites are exposed, and the myosin heads now have a place to grab onto the actin. As long as there's ATP, as long as there's calcium, we will get our contraction. So, now the strength of contraction. It's true that every action potential is exactly the same as every other action potential. And all we can do is send action potentials down the T tubules to open up the calcium. The strength of contraction is a little different. The strength of contraction is based on how many cross bridges can we make in the myofibril. Now, we don't grow new muscle cells, but we can make our muscle cells bigger. We can add more actins and myosins to our muscle cell. The more protein we take in, the more demand we place on our muscle cells, the bigger they're going to get. And so we can control our contraction strength, we can increase our contraction strength by allowing us to have more cross bridges to form. Our myofibrils can get bigger and bigger. We can add more protein. Actin and myosin are just proteins. We can add more protein, and so the myofibrils will enlarge. The muscles will get larger thanks to the presence of the actins and the myosins in the myofibrils. So two things we can control for contraction strength. we can increase our strength by having more cross bridges form. And the more cross bridges will form if we have more myofibrils, if we have more actins and myosins. Now, the number of contracting muscle cells, we can't do anything about the number of cells we have. We can make them stronger. But we can also stimulate more muscle cells for the contraction. The amount of muscle cells you would use to pick up a 1 pound weight is different from what you would use to pick up a 10 pound weight. You would use more muscle cells to pick up the 10 pound weight. Now, so we don't have any new cells, but they can be stronger cells. So the strength of the contraction depends then on how many cross bridges do we get in the myofibril, how many myofibrils do we have in the muscle fiber. That's one thing we can alter both of those without affecting the muscle cell and fiber. We're stuck with the same number of muscle cells, but we can make them stronger. Or we can also stimulate more muscle cells. Hope I'm not confusing this. Just remember that muscle cells are fixed. The number of muscle cells we have is a fixed number. We don't grow new muscle cells. We can make the muscle cells stronger through exercise and protein intake. We can produce more actins and myosins. So we'll have more myofibrils in the cell. So we can make the cell stronger. The other thing that we can do is that our brain, our nervous system can stimulate more, we can use motor nerves to stimulate more cells, depending on what we're trying to do. So even if we didn't get a lot more myofibrils in the muscle cell, we'll just use more cells. We have lots of cells in each muscle. We don't have to, we can use them all if we need to. We can bring in other muscles to help us. So we can stimulate more and more cells too. So. Now, what about, well, what about the contraction of the muscle? Because see, we always have different demands. We have many, many different demands on our muscles. It's not always the same. When you go to pick up a pencil or a computer, your tablet, your notebook, when you go to pick up something you dropped, when you are using you're digging a hole in the ground to put a tree in, you're picking up your child. We're going to use different muscles at different intensities at different times. The force of the contraction, the length of the contraction of our muscles and our muscle cells depends on what the stimulus is. It depends on what the demand is. Again, are you going to spend more energy are you going to have more muscle cells involved picking up a 1 pound weight or a 10 pound weight? So it just depends. We automatically adjust the number of cells that we're going to use based on what we're trying to move. Whether we're picking up the 10 pound weight or the 1 pound weight. How long are we going to have to hold it? Are we going to carry a 25 pound bag of sugar into the house? From the garage into the house, and you have, how long are you going to be able to carry that 25 pound bag up 2 flights of stairs or whatever? Perhaps you got a 50 pound bag of potatoes. I don't know why you'd have a 50 pound bag, but let's say you have a 50 pound bag of potatoes. How far are you going to have to carry that? How much force are you going to have to exert to carry that 50 pound bag of potatoes instead of carrying a 5 pound bag of potatoes? So consider the fact that all of our demands on our muscles are constantly changing, all under the control of the nervous system, but constantly changing. The demands and the forces that we apply are in a constant state of flux. They're always changing. When you're driving a nail, are you going to use the same force to hold the nail that you're going to use to hit the nail with the hammer? You know, are you going to be trying to force that nail into the, are you trying to force that nail into the hammer, or are you trying to hit the head of the nail with the hammer? Which hand are you going to be using more muscle cells? So, okay, every muscle is connected to at least one motor nerve. Every muscle is served by at least one motor nerve. I didn't say muscle cell, I said muscle. Every muscle cell is connected to a motor nerve, but every muscle has at least one attached to it. Now, a motor nerve can form axon terminals that will connect to 300 or 400 different muscle cells. So you can connect, each nerve can have an axon terminal for 300 or 400 different nerves, 300 or 400 different muscle cells. And so the axons with their axon terminals can, you know, one nerve can connect to 400 muscle cells. They're all going to get the same signal at the same time with the same intensity. Do something, maybe, or don't do something. The connection between the nerve and all the muscle cells that it stimulates is called the motor unit. Given that we have a muscle cell, we have a muscle, let's pick a muscle. How about the biceps brachii? In the biceps brachii, we've got thousands and thousands and thousands of muscle cells. And we are innervated. The muscle is innervated by several, at least several motor nerves. Each motor nerve can connect through its axons to 400 individual muscle cells. Each axon, that, you know, all the axon terminals from one nerve and all the muscle cells that those axon terminals connect with are called the motor unit. That means that entire motor unit will get the same, every cell in that motor unit will get the same signal to contract, because that's all muscle cells can do. They can contract. They don't contract weak. They don't contract strong. They contract. There is no such thing as a weak muscle cell contraction. There's no such thing as a strong muscle cell contraction. Again, there's no such thing as a weak action potential or a strong action potential. Once a cell gets an action potential, once a muscle cell gets an action potential, it's going to contract. It's going to contract to its fullest extent every time. So every muscle cell, when it's stimulated, contracts to its fullest extent. So how do you have a weak muscle contraction or a strong muscle contraction? Depends on how many muscle cells you're firing that are being stimulated. Depends on how many muscle cells are being used to do the job. Are you using enough muscle cells to do the job, or are you using too many muscle cells to do the job? Are you using a sledgehammer to drive a nail, or a regular hammer to drive a nail? You know, are you overdoing it? So the motor unit is the neuron, the motor nerve, and all the muscle cells that are being stimulated by that motor nerve. Usually about 300 or 400. Now, if you have a nerve that's stimulating 400 muscle cells, you have pretty good control over those muscle cells. You have general control over the muscle. You're not going to be using that muscle for precise actions. The more motor unit, the more cells you have in a motor unit. Remember, the motor unit is a nerve and all the cells that are being innervated. The more muscle cells you have in a motor unit, the less precise the action is going to be. If you want precise action, you want to have 3 or 4 muscle cells per neuron, like the muscles of the eye or the muscles in our hands. For precise action, the fewer is better. So the smaller, the fewer the cells we have being stimulated by a motor unit, the more precise control we have. Now, if we're dealing with 300 or 400 muscle cells in a motor unit, those cells are not going to be grouped together. They're going to be spread throughout the muscle. So, a couple of reasons here. One, it warms up the whole muscle, and warm muscles work better than cold muscles do. And second, if we stimulate 400 muscle cells, we're going to get an even contraction, even if it's a small contraction of the entire muscle. Now, it's pretty unusual to have one neuron, one motor unit, firing all by itself in a muscle. You know, you're not going to turn on 300 muscle cells in your biceps. Generally, you're going to turn on a lot more than that to do whatever you're going to do. It's entirely possible that you could have one motor unit firing, but usually doesn't work that way. But when a motor unit fires, the entire muscle will contract slightly. We say it's a weak contraction. Yes, it is a weak contraction. Now, every muscle cell in the motor unit contracted to its fullest extent. But if you consider all the muscle cells in the muscle, and they're the only ones pulling on the muscle, then it is sort of a weak contraction because you're not getting, your muscle isn't going to move very far. It's not going to contract very much because you got all the other thousands and thousands of cells that aren't doing anything. So you get a weak contraction. Not because the individual cells aren't trying, they're trying as hard as they can to contract. They're just not enough of them contracting. So here's a motor unit right here. Here we see, here's a motor nerve right here, and we have axon terminals connected to different muscle cells. Each one of these is a cell, and within the cell are the myofibrils, and we have the axon terminals connecting to each of the at the neuromuscular junction at every cell. So these individual cells here are being stimulated by, the axon terminals. Each connection, for this is motor unit 2. It connects to these 3 muscle cells. Here's motor unit 1. It connects to these 2 muscle cells. That's a motor unit. It could be connecting to 300, or it could be connecting to 3 or 4 like you see here. And so the axon terminals connect at the neuromuscular junction. That's part of the motor unit. Every cell in the motor unit will contract when stimulated. How many motor units do we have to bring to play to get something done? Well, it depends. It depends on what we're trying to do. It depends on the stimulus. So now, here's something else to think about. We have something called muscle tone. Now, what happens with our muscle cells to keep them warm, since warm muscles work better than cold muscles do, motor units are going to have a constant rotation of stimulus. So all the motor units in a muscle cell, each individual motor unit, whether you're contracting or not, each individual motor unit will receive a stimulus, will receive an action potential, and that will cause a very, very slight contraction of the muscle. It also warms up the muscle, and then another motor unit does it, and then another motor unit does it. We don't create any kind of movement in the entire muscle. What we're doing is generating what we call muscle tone, where we keep the muscle warmed up. We keep some calcium still available in the sarcoplasm. We keep heat available. We generate a little bit of tension that helps to hold joints together. And we keep rotating this around through the muscle cell. so that each of the cells in the motor unit contract, and then they rest. And then the cells in a second motor unit contract. And then maybe a third motor unit on the other side of the muscle contracts. And then the fourth motor unit, and so on until all the motor units have fired at least once. And then we start over again. It's all random, but it keeps the muscle warm. It keeps the muscle slightly contracted. It keeps a little bit of calcium in the sarcoplasm. And it creates a little bit of tension to help hold the joints together. This is what we refer to as muscle tone. It keeps the muscle slightly firm, slightly tense. This is muscle tone. Now, a great distractor on an exam would say that muscle tone is the pleasant sound your muscles make during exercise. Please don't mark that one on any exam. There is no sound involved with muscle tone. And you know, I wouldn't be saying this if someone hadn't marked that on a test. So anyway, enough of that. So that's muscle tone. Muscle twitch is something that doesn't really occur. It's a theoretical concept. This is a muscle cell contracting based on one action potential from one motor nerve. Because muscle cells, it's extremely rare for a muscle cell to be stimulated, contract once, and be done. We just don't operate that way. Now, it's possible, but we just generally don't, our bodies just generally don't operate that way. We don't have a single contraction from a muscle nerve, motor nerve, telling a cell to contract and then don't do it again. But we call this a twitch. So it is more of a theoretical concept than anything else, but the twitch explains how muscle cells operate. Now let's take a look at this. The muscle twitch has three phases. It has the latent period, when excitation contraction coupling is taking place. In other words, where the muscle cell is receiving its action potential, it's opening up the terminal cisterns, it's flooding the sarcoplasm with calcium. Calcium is binding to the troponin, and the myosin heads are reaching up and grabbing onto the actin and giving it a yank. Well, getting ready to, well, at least binding to the actin. That's the latent period, the buildup, if you will. The period of contraction is when the cross bridges form and the myosin heads give the actin a yank. And then we have the relaxation phase where all the calcium goes back into the sarcoplasmic reticulum and there is no more tension. So we have the action potential opening up the terminal cisterns, the whole depolarization event and everything. Terminal cisterns open up, flood the sarcoplasm with calcium. Calcium binds to the troponin. Troponin rolls out of the way. Tropomyosin goes with it. Binding sites are exposed, and the myosin heads grab onto the actin. We have the contraction phase. The cross bridges have formed. They contract, pull the actins towards the midline of the sarcomere. Then they're going to let go if there's more ATP, and if there isn't, they're going to stay attached there, or they're going to let go, and then we're going to go back to our relaxed phase when we lose all the calcium. Contraction is fast. Relaxation is slow. Contraction is very fast. Relaxation is slow. So these are the three phases of the muscle twitch. We can see just how fast it is. Here's our latent period. This is in milliseconds. This is about 5 milliseconds right here. So this is the latent period when we are receiving the action potential, opening up the calcium filled terminal cisterns, flooding the sarcoplasm with calcium, allowing the calcium to bind to the troponin, causing the troponin and the tropomyosin to roll out of the way, exposing the binding sites. And as we expose the binding sites here, we can see that the tension increases as the myosin heads grab onto the actin. We have our contraction phase of about 10 milliseconds, and then we spend a good 100 milliseconds relaxing. This is a twitch of a muscle cell. A very rapid phase of contraction and a long period of relaxation to get us back down to zero tension. Now, muscle twitches, when they do occur, are not isolated phenomena. They occur repeatedly. They involve many cells. I mean, consider the muscle cells around the eye. You know, we're getting twitches on the left-hand side. The eye muscles are contracting. The eye muscles are contracting rapidly and repeatedly. Now, over on the right-hand side, you can see the thick muscles of the hand, particularly right below the thumb, that is contracting slower. You know, it is a slower contraction rate than what's happening at the eye, and it stays contracted slightly longer. So we don't have an isolated twitch by itself. We generally see lots of muscle cells involved in a twitch because we have twitches all the time. We just don't have a cell doing it once and done. But every time we do have a twitch like this, we're going to go through the latent phase, the contraction phase, and the relaxation phase. Just a lot of cells are going to be doing it. You know, it doesn't take long. The latent period is very, very brief. The contraction phase is very, very brief. Contraction phase can vary depending on the type of muscle cell it is, but it's going to be quick. But we're still going to have a long relaxation phase. So we can see this. Compare in the blue graph on here. So we have our latent period from 0 to 5 for all three of these. The eye muscle contracts rapidly and relaxes quickly because our eyeballs are always moving around. So we have a greater need for our eyeballs to move quickly. So we contract and then we relax very, very quickly within a total of, you know, from the stimulus to the relaxation phase is about 20 milliseconds for us. If we look at the gastrocnemius muscle, we can see that its contraction phase is probably about 20 milliseconds, and its relaxation phase is about 60 milliseconds. And but it contracts very quickly and relaxes quickly. Consider the soleus muscle. It has a slow contraction phase and a slow relaxation phase. 200 milliseconds from start to finish for it to relax. And it takes up to 80 milliseconds for it to contract fully, which to us doesn't mean a thing. To us, it's always instantaneous. We don't say, oh, look, there goes my soleus muscle. It is, oh, now it's contracted. Good, now I can do something with it. Doesn't work that way. 80 milliseconds is, we can't comprehend how quick 80 milliseconds really is. That's 80 thousandths of a second. You know, we can't comprehend something that quick. We can't comprehend how quick the muscles of our eye work or how often they're moving around, or the same with our gastrocnemius. So despite the fact that there are some significant differences here in the speed of contraction and relaxation, to us, it's all instantaneous. But different muscle cells are going to contract at different rates. Okay. Something that we can do, and we do this all the time, since we don't usually operate with an isolated muscle twitch, because, you know, we like to have our muscle contractions nice and smooth, meet the demand. You're not gonna, you're not gonna try and crush somebody's hand when you're shaking hands with them. You're gonna try to be gentle. You're not going to slap somebody in the head you're trying to if you're stroking, if you're holding a newborn, you're not going to be smacking it alongside the head when you're trying to stroke the side of its face, for whatever reason. I don't know. I don't know nothing about newborns anymore. So, but you're going to have your muscle response is going to meet the demand or the need that you want it to do. So, and you want it to be smooth with very little effort. We don't see a muscle twitch by itself. It's just not a phenomenon that we encounter, at least not in healthy muscle. What we do is we have a graded response based on the demands of what we're doing. Consider picking up a 1-pound bag or a 10-pound bag of potatoes. How many are you going to use more muscle cells for the 10-pound bag? Of course you are. You're going to vary your response based on the need. If you go to pick up a piece of paper off the floor, you're going to spend fewer muscle cells than you went to pick up something very heavy off the floor. You're picking up a chair or a piece of paper, which is going to involve more muscle cells. Well, obviously the chair. We meet the need with our muscles. Whatever we're trying to do, we're going to use our muscle cells appropriately to meet that demand. Whatever the demand is, we're going to try and meet that demand. And so we control our skeletal muscles. We control our skeletal muscles so that we can have this graded response. So we have a smooth effort. We have a motion that is not going to have a lot of wasted energy involved or wasted actions. We're going to go and pick up something, or put it down, or we're going to drive a nail, or we're going to press the starter button on our car, or we're going to walk, or we're going to climb stairs, or we're going to exercise, or whatever we're going to do. And we're going to use as much energy as we need to get the task done, and generally not more than that. We're going to have just enough energy to complete the task. That's our muscle response. We base our response according to what we're trying to accomplish, according to what we're trying to move in the environment. Now, there's two ways we can grade our responses. There are two ways that we adjust: by changing the frequency of the stimulation, or by changing the strength of the stimulation. Graded responses allow us to apply any force we need at any time. Within the total strength of our muscles, we can apply any force we want at any time. And to achieve this, we can alter how rapidly we are stimulating a muscle cell. The frequency of stimulation. How often do we stimulate the muscle cell to do something? We certainly do not have a twitch and once and done. We can stimulate the muscle cell repeatedly to do something. So we can stimulate the whole muscle to do something, depending on how many muscle cells are being stimulated. Are we going to stimulate the whole muscle? If we stimulate the whole muscle, then every muscle cell, as always, will contract to its fullest extent. If you bring in all 1,000 muscle, let's say that your biceps has 1,000 muscle cells. If you bring in all 1,000 muscle cells in your biceps, you're going to have a pretty powerful contraction. you bring in 100 muscle cells for your biceps, you're going to have a weaker contraction. You're going to have a weaker force. So how rapidly, how often do you, so you can bring in lots of cells or you can bring in fewer cells. You can stimulate the cells repeatedly to increase your force. So two terms here: frequency of stimulation, how often do we stimulate the individual cells, and recruitment, how many motor units are we bringing together here? If we increase the frequency of the stimulation, if we contract, if we, action potential, one after another, we are increasing the frequency of the stimulation. We encounter a phenomena called wave summation. If we stimulate a muscle cell and then stimulate it again, we get a greater contraction. Because if we stimulate a cell and then stimulate it again, the muscle cell didn't have time to relax. It's warmed up. It also has calcium left over from the first contraction. We stimulate it and stimulate it again. It has calcium left over in its sarcoplasm that never got pumped back in. It has more calcium cut from the second stimulus, and it's nice and warm. Warmer muscles work better than cold muscles do. And if we continue to stimulate it, we will get a greater and greater contraction. Looks like this. Here's what a muscle cell looks like with a twitch. There's our stimulus right there. 0 time. We get our twitch and we get this, we get the level of tension right here at the top of the peak. Then we let it relax and come all the way down here to 200 milliseconds, no tension whatsoever, and we stimulate it again, and our second twitch is just as strong as the first twitch. It's all or nothing event. We stimulate the cell, it contracts, and it relaxes. We stimulate it again, it relaxes. We stimulate it one more time, and it relaxes. And there is no difference in the size of the twitch. The size of the response is exactly the same. It's like the first twitch. The first twitch has no impact on the second twitch. And the third twitch has no impact on the second twitch. And the second twitch has no impact on the third twitch. They are isolated events. But what happens if after the first twitch and it relaxes out to 200 milliseconds, we stimulate it again, and just as we start to relax, we hit it with another stimulus right here at the 100, at the 250 millisecond mark, more or less. We hit it with a second stimulus right here, and we get a second contraction. But we get a greater twitch. There is calcium still in the sarcoplasmic reticulum. Let me rephrase it. There's still calcium in the sarcoplasmic reticulum, but there's also calcium in the sarcoplasm. So there's still some calcium already out there around the actins and the myosins, still binding to the troponin. So now, because we have more binding sites exposed, the myosin heads have more places to grab onto and give it a yank. And so we get a second contraction now that's stronger than the first, because we didn't we didn't attach to all the binding sites the first time. We only attached to the binding sites that were available. But because we now have more calcium, because we started out with, we left some calcium behind, didn't get all the calcium. It takes a while to get the calcium pumped out of the sarcoplasm. There's still calcium there. The calcium, combined with the new calcium coming in from the second impulse, second action potential, fastens on to more troponin, exposing more binding sites for the myosin heads to grab onto. So now we get a greater contraction because there's more myosin heads finding more binding spots on the action. We have more cross bridges forming. We get a greater contraction simply because we have more calcium present. This is increasing the frequency of the stimulus. We've increased, instead of waiting for 200 milliseconds for a second stimulus, we only waited 50 milliseconds. We've increased the frequency of the stimulus. And if we do it again, we'll get a third contraction that's even greater than the first two, and a fourth contraction greater than the first three, and a fifth contraction greater than the first four, and so on. We'll continue to build in here as we find more and more, we have more and more calcium left behind. Every time we stimulate the muscle cell with an action potential, we cause more calcium to leave the sarcoplasmic reticulum, flooding into the sarcoplasm, finding the additional calcium that's already there. So we expose more and more binding sites on the actin. So the myosins have more and more places to form cross bridges, and we get stronger and stronger contractions. And so we keep doing this until we maximize our tension. We call this tetanus, not the disease, but this is tetanus. We're going for what we call or tetanus or tetany. And so we've maxed out the amount of tension that we can generate in the muscle cell. And we will, if we continue to stimulate this muscle cell, we'll reach what we call complete tetanus. The stimulations are occurring so rapidly, the muscle cell is not relaxing at all. And so we now have a smooth curve with no dip for relaxation at all. And so we've maxed out the amount of tension that we can produce. When we are putting forth maximum effort in our muscles, by increasing the frequency of the stimulus to the muscle cell, we will generate what we call wave summation or temporal summation, temporal over time, so that we max out the strength of the muscle. And this is called complete tetanus. Of course, it doesn't last very long because our muscles rapidly fatigue and let go. So, now, let's stop on that one, because I've thrown a lot at you. So, okay, so that's all I've got for muscles today. In lab this afternoon, we're going to take a look at the second part of the general senses. And on Wednesday, we will finish up muscles and hopefully move into the nervous system. So anybody have any questions on anything? Okay, then I'm going to get us out of here, and I will see you in lab this afternoon.